Hello, good morning, everyone. Today we will be doing the test review for test number 1, which is going to be on Thursday. Okay, so you can find a copy of this test review on eCentennial. There should be a link to it in last week's the brief announcement. So here we go. First question. Label the different layers of the fractionated blood in the diagram below and describe components found in each layer. Okay, so if you take a sample of blood and you spin it at very high speed using a centrifuge, you will separate it into the liquid portion as well as the cellular portion. So the liquid portion is what we call the plasma. Okay, plasma. Okay, so I'll try to do this in a more interactive way because it is a review. Can anybody tell me what is found in the plasma?
Waste products.
Yeah, sure. Waste product. So waste such as any specific examples? No? Okay, carbon dioxide, urea, for example. But that's not necessarily the first thing that comes to my mind when we talk about plasma. Sure, there is a little bit of waste, but the major component of plasma would be what? Safi said proteins. Yeah, there are definitely proteins in it. Okay, so what waste product proteins, dissolved ions. Yeah, so dissolved ions such as sodium, chloride, calcium, magnesium, potassium, all these things. Okay, and Mary said water, that is the major component of plasma. Very good. And proteins, right? There are different types of proteins that we talked about. For example, there is clotting factors, right? Clotting factors. Okay, an example of clotting factors would be what and what? Anybody? We talk about two examples of clotting factors in class.
Prothrombin?
Yes, there's prothrombin. And what is the other one?
Fibrinogen.
Yeah, that's correct. Fibrinogen, thank you. Fibrinogen. Okay, so these are necessary for the coagulation to take place. Okay, if you have a wound. And then in addition to clotting factors, we also have like immune proteins, right? Proteins that are important for our immune system. So what are some examples of immune proteins? Anyone want to chime in? Anyone else? What kind of immune proteins are there? Proteins are important for the immune system, either as part of innate immunity or adaptive immunity. What kind of proteins do we have? You can look it up in your notes, right? Okay. Like, what if this was a test question? Where are you going to find the answers? Anyone? Mast cell is not a protein though, right? Mast cell is a type of cell and you wouldn't find it in the plasma. Plasma is the liquid portion. Cellular portion would be at the bottom. Nobody knows any immune protein? You should know at least one. What kind of proteins do we make ultimately to fight infections? Anybody? Yes, antibodies. Thank you. Jane, antibodies. Okay. And we have a bunch of other proteins, right? Such as complement proteins. Okay, so these are also important for immune system, right? Part of your innate immune system. All right. Any question? And the plasma makes up approximately 55% of your whole blood. Okay, approximately 55% of blood volume. So there you have it. That is what the plasma is. Okay. Of course, there are many other proteins, right? We have things like albumin as well, right? Should maybe put it here, right? That is another protein which help maintain the osmotic pressure. Okay. So down here in the middle here, between the plasma and your red blood cells, we have a thin layer. This is the buffy coat, we call it, and that is where you will find your white blood cells plus platelets. Okay, so these are just like approximately 1% of your total blood volume. And of course, there are different types of white cells, right? We have our granulocytes, right? We have our agranulocytes. Okay. We are going to review those later on, but we can list them quickly here. Granulocytes, we have things like neutrophils, basophil, eosinophil. Right. We also have a mast cell that we learn later on. In terms of agranulocytes, we have our monocytes. We have our T cells, B cells. Okay. So these are going to be found right here in the middle. Right? It's like a fluffy layer that just floats in between the plasma and the red blood cell. Okay. So down here we have our RBC. What is another name for this portion? Anybody remember? Start with an H.
Hemoglobin.
Hemoglobin is a protein that is found inside the red blood cell, and that is important for carrying oxygen. Good try. I'm looking for another word that describes this portion. Yeah, Jyoti said hematocrit. That is
Hematocrit.
Thank you, Roseanne. Yeah, hematocrit. Hematocrit. Okay, and that makes up how many percent of our total blood volume? Well, if the plasma is 55%, then RBC is going to be approximately 45%. Okay, I know it doesn't really add up to 100, but you know it's all approximate, right? Okay, so this here, this whole thing, the white blood cells, the platelets, the RBC, collectively we call them formed elements. Okay, formed elements. So a couple of things about RBC. Someone already mentioned that it contains hemoglobin, okay, which carries oxygen. Okay, remember the heart lecture 1, right? That stuff is not on the test, right? So that's only on the lab quiz though. All right. RBC is biconcave, right? Made from stem cells in bone marrow, right? Through hematopoiesis. Okay. It's true for the white cells and the platelet as well. They are also made in the bone marrow through hematopoiesis. What's the lifespan for a red cell? Approximately how many days? Yep, 120 days. That's correct. And where will they be broken down? Broken down at at where? Which organs are responsible for breaking them down? Yep, spleen and liver. Very good. Okay. Of course, the hemoglobin, I'm just writing things out as they come to my mind. Okay, it contains iron in it, and iron is the one that is responsible for transporting the oxygen. Okay. Other things that comes to my mind is EPO, right? EPO, the erythropoietin, stimulates RBC production, right? So if you don't have enough oxygen in the body, then your kidney will release the hormone EPO, which will then stimulate red cell production in the bone marrow. Okay, so if you haven't been studying too much yet, when you study, this is what you should do. Okay, you should take out a piece of paper and then just write down stuff, as they come to your mind, stuff that are related. Okay, so when we talk about red cell, write down everything associated with it. That will help you remember the stuff. Okay, rather than reading the notes, right? You can't read the notes and as the only way of studying. You must write it out. Okay. Any question for the components of blood? All right, let me know when you do. Compare the two test tubes shown below. Explain what might cause the result on the right. Okay, so one of them is normal. Okay, and something is wrong with this one. What is wrong with it? Anybody want to tell me what is wrong with the picture on the right? If I show you this on the test and I ask you, what is the problem with this blood sample? What would you say? There is blood clotting. Could be agglutination. Okay. Now look at the normal plasma here, right? This is the normal plasma. Normal plasma is supposed to be a clear, yellowish liquid, right? Fluid, right? But here is not clear. Okay. It has all these red stuff in it. Okay, so what is happening here? Well, this is actually hemolysis. Okay, remember we talk about like mismatch of blood. Right? If you transfuse the wrong blood to a person, that's what's going to happen. Okay, you're going to have all the red cells rupturing, and when the red cells rupture, the hemoglobin, right, it's going to come out, the protein, right, and it's going to go into the plasma because protein goes into the liquid portion, right? Okay, and hemoglobin is actually what makes the blood red. So by having all these red hue right here, it's a sign that shows you there is hemolysis. Okay, so could be caused by mismatch blood. Okay, so when antibodies bind to RBC, it cross reacts, or you can say a cross reaction occurs, leading to agglutination, man. That's what some of you were saying, right? Agglutination. But this is one step further. Right? After they clump together, the next thing that happens will be hemolysis. Okay, and all the hemoglobin will now be in the plasma. Okay, so that is a really bad sign. If you get a blood sample from a patient and you spin it down, it looks like this, you got to do something about it. Okay, so if you see this picture on the test, remember the correct answer is hemolysis. Next. Match the parents with their babies. Anybody did this yet? Anybody have an answer they want to share? Yes, Patrick, please. Tell me what you got, if you want. Okay, for those of you who have not tried yet, I'm going to give you a few minutes to do this. There could be a matching question on the test that asks you to match the baby's blood. All right. So make sure you know how to do it. Okay, I'll give you a moment. And remember, when you do these questions, you should not be doing any Punnett square or anything like that. Okay. All right, let's take this up then. So remember, there are 4 rules, right? That we wrote down before. I'm just gonna quickly tell you what they are. Number 1, if you are AB, you cannot have type O child, right? Okay. If you're O, then you cannot have AB. Okay. So you should again write these down on the test if you need to. AB cannot have O. O cannot have AB, and I already explained why this is the case before. And 2O is going to be O all the time, and 2 negative will always be negative, right? Okay, and that's all you have to know to do these questions. Okay, thank you for sharing your answers. Several people are sending me the answers. Okay, let's take a look. So first one, A negative and O negative, right? Okay, so if A negative and O negative, then definitely not this one. Okay, this one is positive, right? 2 negatives can only have negatives, so not this baby. All right, and if you are O, then you cannot have AB, so definitely not this baby. Okay, but we have 2 choices. Is it I or is it K? We don't know yet. So I'm gonna put down both, and hopefully we can cross one out later. Is that okay so far? Does everybody understand how I come to this conclusion and how you would be able to do that on your own on the test? If you have a question, please ask now before I move on. Okay. Now, rather than going down the list, you want to find people who have like AB parent or O parent, because those are the easiest to eliminate, right? Like this would be a good one to work on, right? So let's see. One parent is AB, so automatically this is not their baby, right? Because AB cannot have O. And then the other parent is O, then this is not their baby because O cannot have AB. Right? So 2 options are gone now. And then we have 2 more options, either J or K. So which baby belongs to this couple? Is it baby J or baby K? How do you know?
K.
And why is that?
Both negative.
Exactly, right? Both of them negative. 2 negative can only have negative, so definitely not this one. And this is baby K. And since K is taken, then we can cross out K from the first one as well. Okay, so now baby I is taken, baby K is taken. We have 2 more and it becomes quite easy now. Again, we want to do the parents with either O or AB first. If one of the parents is O, then it cannot be AB, right? So that means this is J, and then the last one will be H. Okay, just process of elimination. There's no need to think too much. I hope that helps. Last call. Any question? Don't be shy. If you have a question, I bet someone else in this meeting also have the same question. No? Already. Why not AB negative? Which one? For this one? This one? Yeah, I mean, like the question is, wait, sorry. Oh, this one. Why not AB negative? Because the one of the parents is O, right? If the parent is O, they cannot have an AB baby, right? In order to have an AB baby, you have to pass on either IA or IB. And if you're O, type O, then you only have little I to pass on, right? So that's why if one of the parents is type O, guarantee your child will not be AB. Does that answer your question? Is that okay? To an extent, okay. Can you clarify your question, please? Guys, I'm getting like these private message. That's why it seems like I'm answering a question when no one is typing in the chat. Okay, just so you guys don't think I'm crazy. There's another question here in the chat. If there are 2 negatives, the baby can only be negative. Yes. Okay. If both parents are negative, then the baby is guaranteed to be negative.
Sorry, excuse me. I want to ask one thing.
Yeah.
If parents are positive, then how could the baby could be negative?
Yeah, very good. Okay, that's a common question, right? If 2 parents are positive, how can the baby be negative, right? Okay, it's the same question that people ask about if both parents are A, how can you have a type O baby, right? Remember, it's just because they're positive does not mean they are homozygous positive, right? Okay, someone can be positive / negative and still be positive, right? So if both parents are like heterozygous positive, then if you do the Punnett square, right, you can see there's a 25% chance that they could be negative. Okay. All right. Yeah, sure, please.
For B negative and O positive, can the child also be B negative?
They could. They
Okay.
could. They could. Yeah. Because again, right, this could be like heterozygous situation, right? Like a plus / minus. Yeah. Okay. That was good. If you have any additional questions, feel free to message me. But I'm gonna move on for now. You know, and you know, don't lose sleep over it. Okay, there is going to be at most one of these questions. Okay, so you know, if it really bothers you, just focus your energy on other stuff. Okay. Number four. Okay. Let's try this one. A new blood type called Fluffy has been discovered in humans. There are 3, sorry, typo, alleles in the Fluffy blood group, F1, F2, F3. If F1 is dominant over F2 and F3, and then F2 is dominant over F3, how many different phenotypes are there in the Fluffy blood group? Okay. So let's write out all the possible genotypes first. Okay, what are the possible genotypes here, people? If there are 3 alleles, F1, F2, F3, what are all the possible genotypes? How many alleles can we have again as humans? Two, right? Two. We are diploid. We can only have two alleles, right? Okay. So even though there are three, we can only have two. Right? So what are the possible combinations? Right? We can have like F1 and F1, so 1, 1. We can have 2, 2. We can have 3, 3. Okay, what are some other combinations? Please think about it. Yeah. There could be 1, 2. Okay, there could be 1, 3, and then 2, 3, right? So the total of 6 possible combinations. Okay, these are just like combinations of different alleles, right? If the letters confuse you, right? Just think about like you have 3 colors or something. Okay, like you have red, you have blue, and you have green, right? And you can only take 2 at a time. How many different combinations are you gonna be able to get? You can have 2 reds, 2 blues, 2 greens, right? Red, blue, red, green, and blue, green, right? So that's essentially what this is. Okay, let's talk about the genotype now, the phenotype rather. Okay, so it says F1 is dominant over F2 and then and 3, and then 2 is dominant over 3. So that means when you have 1 and 1 together, this would be like fluffy 1. Okay, 1 and 2 together, because 1 dominant over 2, right? That's what it says in the question. That's going to be fluffy 1 as well. And then that's also going to be fluffy 1. So the yellow one, that represent fluffy 1. Okay. 2, 2 is going to be fluffy 2. Do we have another fluffy 2? Or is that the only fluffy 2? That's right. 2, 3 is also fluffy 2. Well, how do we know that? Well, because it says in the question, 2 is dominant over 3, right? It means when you have 2 and 3 together, 2 is the one that is going to show up. So that is fluffy 2. Okay, and then finally, this one is just going to be fluffy 3. Fluffy 3. So the correct answer would be 3 phenotypes. Okay, there's a similar question, right? In the lecture 2, when we did the poll questions, right? You should revisit those poll questions, right? Because those are like actual questions from the test, right? That I took up, that I took from the test bank. So you should definitely go rewatch some of those things. The one we did in the poll, it was like M and N or something like that, right? All right, let's try part B. If all 3 alleles are codominant with each other, how many different phenotypes will there be? So let's write out the genotypes again. Genotype, which is the genetic makeup. So 1, 1, 2, 2, 3, 3, 1, 2, 1, 3, 2, 3. Okay. So how many phenotype will there be? Okay, let's do a poll. Okay. If you think is 3, please choose A. Okay. And F, not sure. Remember, they're codominant now. All right. I will end the poll. And the correct answer is 6. Okay. So this one and 1, that's just fluffy 1. That's fluffy 2, fluffy 3. And then if it's 1 and 2, because there is no more dominance, right? If you have both 1 and 2 together, then that's going to be like, fluffy 1, 2. It's like AB, right? Blood AB. This would be fluffy 1, 3, fluffy 2, 3. So 1, 2, 3, 4, 5, 6 different types. What would be the answer for 4A? The answer for 4A is 3, right? 3 phenotypes, fluffy 1, 2, or 3. Okay. Any question? No? All right. Here's a pedigree question. Give that a moment. Give it a try. I will take it up shortly. Just want you to give it a try first. Okay, so for something like this, we want to write out all the O's and all the A, B's first, because those are the ones that we know for sure, right? So here's O, and we will put little I, little I. Okay, for AB is IA, IB. Okay, over here, little I, little I. Okay, that's all we know. And for the rest, we will just put down half of what we know. So IB something, IB something. Over here, we have IA something and IB something. Okay. Then we have to kind of logic our way through this, right? So let's start with this one. Okay. Always good to take a look at people with type O blood, and then we can figure out relationship, with the parents or with their kids. So this is type O, which means one of the small I come from the mother, and one of the small I came from the father. Okay, I'll highlight it for you. Okay. That's the first thing we can do. And over here, this I must have came from the mother, because the father can only pass on an A or a B, right? Okay. We don't know which one it is at the moment. We'll come back to it later. And over here, the small I, right? Where did the small I come from? Again, we're not entirely sure at this point. It could come from this parent, it could come from the other parent. We don't know. We have no idea. What we do know is the other allele for this person would have to be a B. Okay, because both parents are type B, right? So it has to pass on a B to this particular individual. All right. Now you might be wondering, well, what if they are both like small I and small I? Wouldn't it be possible for this person to be type O? Yeah, that's possible, but you also have to look at the next generation, right? Okay, you also have to look at the next generation. If this person is type O, right, then it's not possible to have both type A kids and type B kids, right? The fact that he has one of child who is a type B, right, tells you he himself must be a type B person. Okay, so this B is responsible for this child. Okay, which then tells us over here, you know, did the mother got the A from her father or the B? Well, it has to be the A, because otherwise you wouldn't be able to have a type A child, right? Okay. So based on that logic, the father has to be IB small I, and the mother has to be IA small I. Okay. Any question? All right, here's a multiple choice question. Go ahead. Anyone else? Only 6 people? Okay, well, let's talk about this. A drop of Sarah's blood was mixed with anti-A. Okay, so we have anti-A here, we have anti-B here, and then we have anti-RH, right? What happens is clumping was not seen in any other test. So no reaction here, no reaction here, and no reaction here. Based on that result, what blood type is Sarah's blood? Anybody know? Yeah, Megan said O negative. That is correct. Okay, O negative. So let's take a look. The test must have been defective because clumping should have been seen in at least one test. No, not true. Right? As we can see here, if they're all negative, then that means the person is O negative. None of the antibodies in Sarah's plasma reacted with the antibody. That is not true. We are not trying to see if the antibody reacts with Sarah's plasma. We're trying to see if the antibody is reacting with Sarah's RBC, right? Okay. RBC is at the bottom, right? When you spin it down, it has nothing to do with her antibodies in the plasma. That is incorrect. Sarah's blood is O positive. No, we figure out it's O negative, right? Because there's no reaction with the RH. That's why. And D, Sarah does not have any ABO or RH antigens on her RBC. That's the correct answer. Okay. Because she does not have A, she does not have B. That's why we didn't see any reactions with the antibodies. And there is no RH on it as well. That's why there was also no reaction with the RH. So the correct answer is D. One person got it right. Good job, whoever you are. And for those of you who chose C, do you understand why D is the answer? Okay. Any follow-up questions? Okay. Let's try another one. Go ahead. Okay, I'll explain the last one again after we finish 7, okay? Okay, I'm not sure if people are just not here or like you're not sure what to choose. But I'm only getting 5 answers, and it's it's already 2 minutes. So I'll take it up. Okay, so all the safe blood transfusion. Okay, remember to use this chart, right? Okay, and of course, negative can be given to positive. So over here, right, O to A is okay, but positive to negative is not okay. So that's not the answer. Okay, AB negative to AB positive, that's okay. Now you can always give to your own, and negative can be given to positive. A positive, AB positive, also okay. B negative to AB positive is okay. O positive to O negative is not okay because positive cannot be given to negative, right? Okay, so the correct answer is B, C, and D. Okay, Patrick, you asked me to explain this further. Is there a particular part that you need me to clarify? Like, is one of these statements not clear? I guess I'm just not sure which part you want me to elaborate on. Why is it the last answer? Okay, so do you agree that based on the test result, her blood type is O negative? That's the first thing we have to sort out. Do you agree that if there is no reaction with A, B, or RH, then her blood type must be O negative? Yes or no? Okay, so you agree it's O negative. Well, what does O negative means? Okay, if you think about the RBC, okay, if you have antigen A on it, then that makes you type A, right? If you have A and B, that makes you AB. So what makes you type O? Well, if you don't have A and don't have B, then it makes you type O, right? So that's why Sarah does not have any ABO antigen on her RBC. Okay, and think about RH, right? What does RH negative mean? RH negative means, right, there is no RH on the surface, right? So that's why no ABO, no RH. That makes her, O negative. All right. Let me know if you have more questions. Okay, we're going to move on to the next part. Fill in the following chart. Okay. Classification, main function. All right, so macrophage. What is a macrophage? How can we classify the macrophage? Well, the macrophage can either be classified as a phagocyte, right, a phagocyte. It is also an APC, right, antigen presenting cell, and it is also a granulocyte. Okay. What does it do? It does phagocytosis, goes around and eats stuff up. It's able to recognize PAMs, these pathogen-associated molecular patterns. If something has a PAM on it, macrophage is going to go eat it. Okay. As an APC, it is also responsible for activating naive T cell. Okay. And it's able to do that because it can do antigen processing as well as antigen presentation. Okay. That's macrophage. Of course, they came from monocyte, right? Okay. Differentiate from monocytes. All right. Next, we have neutrophils. Neutrophils is a granulocyte. That means you can see their granulotes. Okay, it is also a phagocyte. Okay, the most abundant white blood cell. And what they do is phagocytosis. They just go around and they eat. Okay. Mast cells and basophil, these are granulocytes. Okay, the difference is that mast cells are anchored to tissue. Right? These are anchored to tissue. Gives you like local inflammatory response, while the basophils, they are in circulation. Okay, they move around. If you have like a massive allergic reaction, that's going to be caused by those basophil. Okay. And the main function is to initiate inflammation, okay, by releasing, what's the chemical that is responsible for triggering inflammation?
Histamine.
Histamine, that's right. And of course, the histamine is going to result in vasodilation and increase vessel permeability, right? Okay, so causing more blood flow, things are going to be oozing out, and then they give rise to the characteristics of inflammation: the redness, the pain, the swelling, and the heat. Okay. And again, mast cell, probably should do in a different color. Mast cell is more like local, and the basophil is more like systemic. Okay, like bodily. Okay. Like seasonal allergies, that kind of stuff. All right, NK cells. NK cells. What are NK cells? NK cells are part of the innate immunity. Okay, innate immunity. Okay, and their function is to recognize and kill infected cells or cancer cell, right? Okay, the natural killer cells, that's what it stands for. So these guys, these are part of your innate immunity. Okay, these are the ones that you're born with, right? Your first line of defense. They are usually immediate, do not have any specificity. They are have also have no immune memory, right? Okay, but they work right away. You're born with it. Any question? Of course, this is just a summary, right? Like for the detail, the explanation, right? Look at the study guide that we did, right? Okay, so helper T cells, this is going to be part of cell-mediated adaptive immunity, right? Change the color. Okay, part of cell-mediated adaptive immunity. Adaptive immunity is immunity that you acquire throughout life, right? Through exposure to foreign antigen. They are specific, they are diverse, right? There's immune memory, right? And it takes some time for it to start to work. It's not immediate. Right? So there are two branches of adaptive immunity. We have the T cell department and then the B cell department, right? So everything that has to do with T cells is what we call cell-mediated adaptive immunity. Everything that has to do with the B cell, that would be the antibody mediated, right? Okay. So what does helper T cell do? Helper T cell will activate a naive B cell. Okay. And you know, they coordinate immune response. Whereas the plasma cell, this is part of what did I say? Antibody mediated adaptive immunity. Okay, and the main function of plasma cell is to mass produce antibodies. All these antibodies will be in the plasma, right? When they are produced. Obviously, there are other types of T cells, right? And B cells. There's the memory T cells. There is the cytotoxic T cells. All right. So feel free to add them to the list if you want. But we're gonna stop here. Okay. Multiple choice question again. Please try to participate. It's very important for your sake. Oops. Relaunch. We're doing question number 9. Anyone else? All righty. I will end the poll in 3, 2, 1. Okay, let's see. First one, specific immunity works only after non-specific defense have failed. At first glance, that might appear to be true, but not really, right? Because many, even though when we explain the concepts in the lecture, we talk about specific immunity first, and then we talk about non-specific defense. Actually, both of them can happen simultaneously, right, at the same time. So one doesn't necessarily have to follow the other. So that's not true. Non-specific defense and specific immunity require prior exposure to be effective. Effective. Not true. Okay. No prior exposure is necessary for non-specific defense. B cell works in both non-specific and specific defense. No, B cell is for specific immunity only, right? Same thing with T cell. T cell is for specific only. And so E is the correct answer. Okay. As you get exposed to different antigens, your specific immunity would evolve, would change. You would be able to have more different, more types of memory cells, for example. Whereas non-specific defense, right? Your mechanism for inflammation, fever, right? Phagocytosis, those don't change. Okay, it doesn't change throughout your life. Correct answer is E. Okay, let's try another one. These are like examples of harder questions, right? Like you're not all of the questions are gonna be like this lengthy on the test, right? So don't get too worried, I guess, but be worried enough that you will study it well. So it looks like we are kind of all over the place here. Let's take a look at this question. First one, a RhoGAM injection is an example of artificial active immunity. First of all, active means you have to make your own antibody, and passive means someone else makes it and then give it to you. Okay. Artificial usually involves some kind of injection. Natural does not. All right. So RhoGAM is actually nothing to do with acquired immunity. RhoGAM is a shot you give to RH positive, a negative mother who's carrying a positive baby, not to boost her immunity or anything, but to prevent sensitization, right? So these two are automatically not the answers. Nursing an infant to provide antibodies in breast milk is an example of natural active immunity. Breastfeeding is natural, but the antibody is being passed on from the mother to the child. And as such, this would not be an active form of immunity. Okay, the baby did not make the antibody itself. It's just receiving it from the mom, so it's going to be passive. That's the correct answer. Catching COVID-19 and recovering from it should give you natural active immunity. Contraction is natural. You make the antibody yourself and recover from it. That's why it's active immunity. So this is the correct one. Receiving the vaccine would be an example of artificial active immunity, okay? Receiving the vaccine cause your immune system to make its own antibody and its own memory cells. That's why it's active, okay? It's different than receiving a serum. A serum contains the antibodies, which would be an example of passive. Okay, so this was explained in quite a bit of detail, both in the lecture as well as the tutorial for week 3. So be sure to check it out if you need a refresher. Questions? Okay. Name the cell. Uses porphyrin, which is a protein that put holes in cells. And what cell use that? It is the killer T cell, also known as cytotoxic T cell. And so that could be a multiple choice question, right? Which of the following cell uses porphyrin to put holes in infected cells? And then they give you a bunch of cells. You have to know to choose this one. Next one should be easy. Release histamine to initiate localized inflammatory response. That would be mast cell. A type of APC that activates naive T cells. We only learn one type of APC. That's macrophage. Kills cancerous cells. That's the NK cell. Forms blood clot. That would be platelets, or like the fancy word, it's thrombocytes, right? In case you see it, that's what it means. You don't have to Google it on the spot. Most abundant WBC, that's the neutrophil. That's it. All right, moving on. Formed elements. Okay, so formed elements are include RBC, right, the erythrocytes, the WBC or leukocytes, and the platelets, right, the thrombocytes. Granulocytes, we list them earlier. Neutrophils, basophil, eosinophil. Right? Eosinophil fights the parasites, right? Lymphocytes, we have T cells and all the different types of T cells, and B cells. Coagulation proteins, we talked about this earlier. Prothrombin. What's the other one? Fibrinogen. Autoimmune diseases, that's when your immune system attack yourself, right? What are some examples that we talked about in class? Or any examples that you know? It's fine. Give me some examples, please. Yeah, sure. Type 1 diabetes. Okay, not type 2 though. Okay, type 1 diabetes. That's when you attack the insulin producing cells in the insulin. Not what we talked about in class though, but nevertheless correct. Was there another one? Yeah, rheumatoid arthritis. Rheumatoid arthritis. That's when the white cells attacks the cartilage in the joints and causing deformation of your joint. Very painful. Megan says MS, multiple sclerosis. That's correct. That's another one. Okay, so these are autoimmune diseases. Physical barrier, again, a bunch of them. Right? You can say normal flora. Right? mucous membrane, intact skin. Okay, example of immunodeficiency. And there is primary immunodeficiency, and then there is secondary immunodeficiency. Primary is when you're born with it. And then secondary is when you get infected with it. So give me an example. We talk about 2 examples in class for immunodeficiency. What are they? You should know it even if you should know at least one, even if you have not watched the lecture recording. Immunodeficiency. Not anemia. Anemia is it's like hemoglobin deficiency. It has nothing to do with your immune system. Yeah, Patrick says AIDS. That's correct. Okay, so that is secondary, right? It's acquired, right? Okay, it's something that you catch after you were born. It's not genetically. It's not a genetic condition. Okay, and then the other one that's genetical, that's genetic, is SCID. Okay, severe combined immunodeficiency. You're born without an immune system. The bubble boy, right, that we talked about. Lymphatic organs, there are a bunch that you can have here. Spleen, excuse me, spleen, thymus, tonsils, right? Appendix, what have you, a bunch. Artificial passive immunity, we talk about serum. Natural passive immunity, that would be like breastfeeding. Okay. Characteristics of inflammation: redness, swelling, pain, and heat, right? Warmth. All right. Why do we need to receive a flu shot each year in order to have protection against flu virus? I think we talked about this before, right? Is it A, they are all booster shots to strengthen the immune system? Each year, the dominant strain of flu circulating changes, right? We talk about this in the lecture recording, right? Remember? We don't know, there's so many different strains, right? The H and the N changes, and, you know, we have to create a new one. 14. Okay, let's try this one. Did we do this? I feel like we have. I don't remember. Sorry. All right. So we're gonna take some T cells out. We're gonna, expose it to some antigens, and we're gonna put it back in. And in order for this to work, what do you need it to happen? Or like the question says, for this to work, the receptor must do all of the following, except what? Okay, so let's see. Recognize an antigen that is found only on cancer cells and not other bodily cells. That one we want it to happen. Okay, you do not want a T cell that would not be able to distinguish what's cancerous and what's not. So this is something that we do want to happen. So that's not the exception. Recognize an antigen that constantly mutates in the cancer cells. That actually is the exception. Okay. Remember, the antibodies that we can produce is only good for one type of antigen, right? So if there is something that keeps on mutating, keeps on changing, like the flu virus, for example, then the antibody that you produce the first time will not work for the second or third time, right? It's impossible for you to have just one antibody that will work for all the kinds of different mutations. So this is the exception. Okay, you wanted to trigger clonal expansion. Remember, once you selected the correct cell, you want it to divide into multiple copies of itself, and then they differentiate into the different effector cells, right? So that's necessary. And then also, produce effector cells that would attack cancer cells, right? Of course, that's also needed. Okay, so it has been, actually, you know what, we're almost there. So maybe we just skip the break and we will just soldier on for another 15 minutes. Okay, next part. Answer the following questions. What is the expiratory reserve volume of this person? Okay, so we have to know how to read the graph. And expiratory reserve volume is right here, right? This portion is the expiratory reserve volume. So if you take a look at here, this is 3,000, this is 2,000. So I will say this is like about 2250. Okay, what was the answer for 14? For 14, the answer is B. Right? This is the exception. We needed to do this, but we cannot make it do this. Okay, because if it keeps on mutating, it's impossible to have one antibodies for it, right? It would be ideal if we could do it, but we can't. Okay, so this is 2,250, I'll say, and then down here that would be like 1,250. So if you subtract it, if you take this minus this, it will be 1,000 milliliters. Okay, on the test it will be more clear, the graph. Okay, like I mean, this is kind of clear, I would say, right? 2,000, 1,000. So halfway is 1,500, and then half of a half is 1,250. Okay, but it's not going to be this particular graph. It's going to be more clear on the test. What is the label for B? Okay, what is B? Anybody from this point all the way to this point? What do we call that? Anybody remember what this is called? That is your vital capacity. That's correct. Thank you. So vital capacity. And just to refresh your memory, the vital capacity is inspiratory reserve volume, which is C. Okay, C plus tidal volume, which, do we have a label for that? I don't think we have a label for that. That's the tidal volume, okay? And then plus expiratory reserve volume. Okay. Describe the breathing pattern for the person. Okay, so I'm just gonna describe it to you. I'm not gonna write it out. Okay, so this is regular breathing, just breathing in and out and in and out. So the lungs expand as you breathe in and contracts as you breathe out. Breathe in, breathe out, breathe in, breathe out, breathe in. And then at the end of the exhalation, you exhale as much as you can. So it takes you all the way down to this point, okay? And then you breathe in again and out and in and out. And then here, you breathe in as much as you can until you can't do it anymore, Okay, and normal breathing again. And then again, you inhale as much as you can. And then from this point, after your maximal inhalation, you breathe out as much as you can. So all the air that came out, that would represent your vital capacity. Okay. Describe the volume label as A. A is the residual volume. We talked about residual volume before, right? It is very high in carbon dioxide. It's the so-called dead air, right? Does not involve in gas exchange. For people with emphysema, we talked about that last week, the residual volume would be higher, right? Because of all the empty spaces created by the alveoli merging together. Okay. Next, draw a flow chart to illustrate how inhalation and exhalation occurs. Okay, we kind of did this last week, but let me just do a quick one here for you. Inhalation. So in order for inhalation to occur, your rib cage moves out and up. This is facilitated by your intercostal muscle, right? The muscle that moves your rib cage, pulls it out and pulls it up, right? And then simultaneously, the diaphragm contracts, flatten, and moves down. This is not really a flow chart, I guess. Here I put an arrow, it makes it look like a flow chart. Okay, so together, these two actions, remember the picture we drew last time? The picture we drew, this is an exaggeration, but it helps you remember it. Ribcage is going to go out and up. Diaphragm is going to go down and flattens. And because the lungs have the pleura, right, the double membrane, right, the movement of the lung is tethered to the movement of the rib cage and the diaphragm. So if this moves out, it will pull the lungs with it. If the diaphragm moves down, it will pull the lungs down with it as well. So collectively, well, I should say the movements of the lungs are tethered to the movements of rib cage, and diaphragm. Okay, because of the pleura and the pleural fluid, right? That's why in pneumothorax, when you puncture your lungs, okay, your chest will move up and down, but your lungs will no longer move with it. So you cannot breathe on the side with the puncture lung, a puncture pleura. Right? So collectively, what's going to happen? Well, because rib cage goes out and up, diaphragm comes down, you will increase the lung volume. And then because of Boyle's law, the pressure is going to decrease. This is Boyle's law. And then finally, when PI, the pressure in the lung is less than PA, right? Air rushes in. This is because of pressure gradient, right? Pressure gradient. Okay. Fluid tends to go from high pressure to low pressure. So if the atmospheric pressure, the PA, is higher than what's inside the lungs, then the air goes in. Okay, so that's for inhalation. And of course, exhalation is the other way around. Right? I'm sure you will be able to write it out yourself. Any question about this? Okay, so just a couple more multiple choice here. During swallowing, what happens? Okay. Larynx moves upward against the epiglottis. Now, that's the correct one. Okay, you can feel it. You can put your hands here in your neck and you swallow. Like that, you can feel something is pushing up, right? So this is your epiglottis, the little elastic flap. And when you swallow, the larynx goes up and it gets sealed, right? Prevents choking. We saw that in the X-ray that I showed you last week, right? Surfactant. Okay? Surfactant is the oily substance produced by these septal cells in the alveoli, right? That breaks down surface tension. Right? So surfactant will reduce surface tension, decrease surface tension to allow inhalation. We talked about babies who are born prematurely. They don't have their own surfactant yet, making it difficult to open up the lungs, right? There is too much surface tension. So that's why you have to inject the steroid through the nose to break the surface tension to help them breathe. Okay, number 19. I expect everybody to get 19 right. We talk about this multiple times. So please choose the right answer for me. All right, and the correct answer is increase in carbon dioxide level. Okay, you have the respiratory center is at the brainstem, right, in the medulla oblongata. Okay, that's where the respiratory center is, and it detects the amount of carbon dioxide in the blood. If it's too high, then you are going to have the urge to breathe. All right. So that is it for the review. I will give you like a link to a Kahoot if you want in the chat box. Oops, I only sent it to one person. Give me a second. There we go. I'll post it on eCentennial as well, but then you know, I have to wait for the video to get processed, so it might be a while. And if you want to do it, the Kahoot thing now, you can do that. But yeah, that's it. Is there any other question that you might have before we end today's session? Anything that you want me to talk about in preparation for the test, which is on Thursday, right? Of course.
I have a question, Professor.
Yeah. Okay. Patrick first, and then your team. Go ahead, Patrick.
So, you know how the quiz is out of 2 attempts? So like
The
is
best
it-
one counts. Yeah.
best one. Okay, thank you.
For the lab quiz. Yeah, he's talking about. Okay. And the student had a question. Yeah. Go ahead.
For the lab quiz, is that timed? It was timed. I thought you said unlimited.
I'm sorry, you a little bit cut off there. Can you repeat one more time, please?
Sorry. For the lab quiz, is that timed?
Yes, it's timed, but it's like an hour or something. And there's only 20 questions or so you have a lot of time. Yeah.
Okay, and then the second question is, where should I find the answer for the answer for last week.
The answer for the study guide?
Yes, for last week.
Yeah, if you go to the homepage, you know how I post a debrief every week? Here, let me show you.
Yes.
quickly. What do you guys are okay? Hold on. Screen one right here. Okay, so the debrief, it says like tutorial archive. That's where you can find a video, and then study guide answer. You just click on that. Okay. You see that? It will take you to
Yes,
all.
thank you.
Okay, no problem.
Perfect. Thank you very much.
Okay. Anyone else have anything else? If not, I mean, you can always email me. I reply you quite quickly. Yeah, good luck. And I will see you next week for the tutorial. Okay. Have a good week. Bye, guys.